Hello friends, today we are going to study enzyme that we can use in recombinant DNA technology. Now, what is recombinant DNA technology? In recombinant DNA technology, we can either isolate a particular gene or we can modify a particular gene in an organism, or we can replace the faulty gene with the normal gene in an organism so that the new characteristic can be introduced into the organism. Okay, suppose a person has faulty gene for a particular protein. Suppose that is for this insulin. Okay, so we can modify that gene in an organism, and we can replace that faulty gene with the normal gene. Okay, so in this way recombinant DNA technology works. Means it is a technique that can modify a particular gene, or that can isolate a particular gene, or make copies of a particular gene, many copies of particular gene, so that more product of that gene can be obtained. Okay, so let's move ahead. So today we are going to study enzymes that we use in recombinant DNA technology. Okay, so let's start from first enzyme, that is DNA polymerase I. Now, DNA polymerase I has, you should remember what are the properties of this enzyme, because if we know the properties of this enzyme, then we will know where we have to use this enzyme. So it's very important to know what are the properties of these enzymes. Okay, so first one is DNA polymerase I, and the properties are: it has 3' to 5' exonuclease activity. I will tell you what is exonuclease, what is polymerase. First, just know the properties. 3' to 5' exonuclease activity, then 5' to 3' exonuclease activity, and 5' to 3' polymerase activity. Now, what do you mean by 5' to 3' exonuclease activity? Means in 5' to 3' exo, as we know, DNA has two sides. One is 5', other is 3'. Okay, now 5' to 3' exonuclease activity means from 5' it can remove nucleotides. Okay, from 5' direction it will remove nucleotides towards 3' direction. Okay, and 3' to 5' exonuclease means, okay, 3' to 5' means from 3' direction it will remove the nucleotide. Exonuclease means removing of nucleotide. Okay, and polymerase activity means it has 3' to 5' polymerase activity. Then what is polymerase activity? Polymerase activity means addition of nucleotide. Now this is one gene. Okay, this is a particular DNA. Okay, 3' to 5' polymerase activity means addition of nucleotide in the 3' direction. Means this enzyme can add nucleotide in the 3' direction. Okay, so there are three properties of DNA polymerase enzyme. First one is 5' to 3' exonuclease activity. Second is 3' to 5' exonuclease activity. Exonuclease means removal of nucleotide. Okay, and polymerase activity means addition of nucleotide. Okay, 3' to 5' polymerase activity, it means, sorry, so sorry, 5' to 3' polymerase activity. 5' to 3' polymerase activity means addition of nucleotide in the 3' direction. Okay, so exonuclease means removal of nucleotide, polymerase activity means addition of nucleotide. Okay, so fine. let's move to our next enzyme, that is reverse transcriptase. Okay, now reverse transcriptase is also known as RNA directed DNA polymerase. Okay, so what reverse transcriptase do is, suppose we have the single stranded DNA. Okay, so from DNA we can make RNA, okay, by using reverse transcriptase enzyme. Okay, so to remember this, just remember, from DNA if we want to make RNA we can use reverse transcriptase enzyme. Okay, always. Now let's move ahead to our third enzyme, that is known as Taq DNA polymerase. Okay, now Taq DNA polymerase is very well known enzyme, as we use this enzyme in PCR method. This enzyme best works at 72 degrees Celsius, and the properties of this enzyme is it has 5' to 3' polymerase activity. Okay, and it also has 5' to 3' exonuclease activity. As I have already explained what is exonuclease, what is polymerase, so you people might know now. So this is the properties of Taq DNA polymerase, which is very important, as we use this in PCR method, and PCR is very important technique for the amplification of DNA, and we use this method in molecular biology techniques. So today we have learned three enzymes. First one is, I will just recall once everything. First one is DNA polymerase I, which has 3' to 5' exonuclease activity, 5' to 3' exonuclease activity, and 5' to 3' polymerase activity. Okay, then next one is reverse transcriptase, which we use to make RNA from DNA. Okay, then third is Taq DNA polymerase, which has 5' to 3' polymerase activity and 5' to 3' exonuclease activity. Okay, so these are the three enzymes that we use in recombinant DNA technology. So this is it for today. Keep learning. Thank you so much. Keep sharing the knowledge. If you have any doubt, please comment. Thank you.